Okay, so we should be recording. Yeah, so thanks for coming. We have Brittany Story from University of Tennessee Knoxville speaking today about internal navigation and topological data analysis. So go ahead and get started.
Awesome. Alex, thank you so much for having me, and thank you all for deciding to attend this class, even though I know your class is asynchronous. So as you'll see, my Zoom name says Brittany Carr. That's just because I haven't figured out how to switch it on Zoom. But my name is Brittany Story, and I'm a postdoc at the University of Tennessee Knoxville. And I actually live and work out of Maryland because I am in a collaboration with the Army Research Lab out of Aberdeen Proving Ground up here. So some of my work will all talk a little bit about how we're going to apply this to outside things instead of just looking at the neuroscience aspect of it all. So to give you a quick outline of what we're going to talk about today, we'll have a little introduction into animal navigation. We'll look at the neuroscience behind the navigation. And then a natural question to ask in some sort of math class is, where does the math fit into all of this? So we'll talk about that a little bit. And we'll look at embedded manifolds of mammalian navigation. And then I'll talk a little bit about the work that I've been doing with one of the graduate students, Eddie Mitchell, out of University of Tennessee, and our work on something called the simplicial convolutional recurrent neural network. So we'll end with a summary like all good talks do, and then I'll talk a little bit about our future work and where we're going. Okay, so let's start basic, and let's just look at a few different kinds of navigation. So our traditional quote unquote navigation is something with a map and a compass, right? So if you know where you are in a location and you have a map and you have a compass, so you have your orientation, your direction, and then you also have your current location and where you want to go, then you should be able to navigate from point A to point B as long as you kind of know a few things. So either you know your speed, you can see where you're going, right? Something like that. So you have some sort of reference that you're able to navigate throughout your environment. So the navigation that's a little more new school is GPS navigation. So, right, if you have your phone or some sort of device that has GPS enabled, then basically you have a bunch of satellites over you in a network, and your signal is going up, down, and it's getting crossed enough. And if you're positioned within the ring of 4 satellites, then you can have a pretty accurate location of what your location is relative to your space. So that's all well and dandy, but we also want to be able to navigate within our own environment, right? So if you're in your house, your office, you want to be able to get to the restroom, the kitchen, all of those good and important things. And we don't really use GPS or a map or a compass. We use some sort of internal navigation. So we know where we are, we know where we want to go, and we kind of know how to get there. And if we don't, humans are pretty good at figuring it out. So that's really what we're going to be talking about today. And that's the navigation within one's environment, or this thing that I'm going to call internal navigation. So internal navigation, as we just discussed, that mammals need to be able to map their environment in order to function. So this includes familiar environments, so like your home or your work, as well as unfamiliar environments, right? If you're in a new city, you want to be able to get from point A to point B using whatever transit mode you choose. So we have some inputs into our body that allow us to kind of capture this information. So we have our typical 5 senses, right? The one we most commonly use is probably sight and hearing. We have some balance information from the inner ear, right? So that you're actually able to walk. We have orientation, right? Which direction you're facing. And then we also have body awareness. So for example, if I close my eyes, I can touch my nose even though I can't actually see it, right? So you have a sense of where your body is in space. And so, right, whether we're in some sort of city environment, nature environment, or in some sort of like inside environment, we're able to get from point A to point B using these senses that we're equipped with. Okay, so a natural question is how does the brain accomplish this navigation? So like I mentioned on the previous slide, we have certain inputs, so environmental information from external sources, so the sights, the sounds, the direction you're facing, etc. And you have some internal processes. So something goes on in your brain that says, okay, I'm taking in all of this information, I'm having neurons that are going to fire, and then the output is going to be knowledge about the information of the space that I'm in. So for example, one piece of information might be the size, right? How big is your room? Is it really small like a bathroom, or is it large like a giant gymnasium? You might have location of important resources, so like access to food, water for, basically all animals, and where you are located in the space. So in the picture that we have below, right, our stick figure says that I'm in the far side of a medium-sized room relative to the window, facing the window about 4 steps from the couch and coffee table, in the middle of the front and back walls. Right, so very rarely do you actually think about all of that verbatim. Instead, you kind of just know where you are in space and where you need to get to go. So there are a few types of cells in your brain, and that's what we're going to mainly look at, that allow this navigation to happen. So we have things like border cells, speed cells, place cells, head direction cells, grid cells, and conjunctive cells. And the main ones that we're going to focus on are the bottom 4. So I'm going to walk through all of them just to give us the same language to talk about with all of the different types of cells. And I'll give you a quick intro to the top 2 just so that we know what's going on in our brain. But before I get started, does anyone have any questions? Okay. In that case, we'll keep cruising. And I normally say this at the beginning, and I think I forgot, but feel free to ask questions at any time. Just unmute yourself and ask. I love having conversations, so this is a great way to do it. Okay, so the first cells we'll talk about are border and speed cells. And for border cells, right, you can look at a wall and your brain automatically is like, oh yes, that is a wall. I do not want to walk into it. That would be bad. And they give you kind of a reference point for where you are in your space. So when your input, your sensory input, in this case, we'll say sight, sees a border at a specific distance and a certain orientation, there's a certain cell in your brain that will fire, right? And it will fire anytime it encounters that kind of environmental stimulus. So these cells are also called boundary cells, and they're found in the hippocampal formation and a few in the MEC, so the medial entorhinal cortex. Ugh. So similarly, right, you also have cells in your brain that fire when you are walking or moving a certain speed. So these are found in the entorhinal cortex, and the firing rates are determined by the animal's speed, and they're independent of visual cues, right? So even if you were to walk around in darkness, you still have some sense of how fast you're moving just based on, what your body is doing. So, okay, we have our walls, we have our speed, so we're getting closer to be able to do some sort of navigation. And so next we'll talk about place cells. So place cells are found in the hippocampus, and these cells specifically fire when you're in a certain location in a certain environment, right? So maybe because I sit here at my desk, probably more than I wish I did, then I have a cell in my brain that fires and says, okay, you are in your workspace, something like that. So these cells remap based on environments. So if you're in a different environment, it is possible that that cell will fire if you're in a specific space in that environment, right? Because that's useful for your brain. Your brain doesn't want to waste resources on cells that only fire in one place and one time forever, right? That would be very wasteful. So they're potentially used to map relevant places in the environment. And I will note that place cells are slightly controversial because a lot of the testing that scientists have done have been on rats in like very specific test environments. So like some sort of box, or some sort of grid navigation space. And then I read a paper recently that they also could just be a type of memory cell, right? Because your memories can be attached to the environment that you're in. So if you go to the coffee shop where you had the best latte of your entire life, right? That might be something that your brain wants to capture. So they're very popular to hear about in the literature, but just saying, there is a little bit of controversy, like any good scientific theory. Okay, so next we'll talk about head direction cells. So these are actually found in a lot of different brain regions, such as the cortical regions of the post-subiculum. The biology words are hard for me as being a traditionally trained mathematician. And then we also have the retrospinal cortex and the entorhinal cortex. And so head direction cells fire when your head is facing a certain direction. So this does not matter which direction your body is facing, right? So if I went ahead and I turned my body this way, then my cells that fire when my head is facing this way are going to fire no matter the way that my body is oriented. And I do want to point out that that is not as easy a mapping as saying you have one head direction cell that fires when you look straight ahead, right? There's clusters of cells that fire for some given direction. So you have, right, if you have a bunch of them throughout your brain, then you have some sort of kind of, I'll call it triangulation, but you have these cell head direction fields that overlap, and when enough of them overlap in the right way, that's when you get the corresponding head direction. And so this is one of the cells that we're going to use in our research, so I'll definitely talk about it a little bit more as we keep moving forward. Okay, and last but not least, are grid cells. So grid cells are kind of the, I would say, super spicy cells because they're relatively new as far as when they were discovered, and some of the people who discovered them actually ended up winning a Nobel Prize for it because it was kind of a big deal, right? Because these were the cells that really kind of unlocked how mammalian brains are able to navigate through an environment. So these grid cells are found in the entorhinal cortex, and what they do is they create multiple sized hexagonal grids that overlay your environment to do something like triangulation. So let's look at this upper left-hand picture right here. So what this is kind of expressing is that if I am in any one of these locations of these dark clusters, right? So I could be here, I could be down below, I could be anywhere on that hexagonal grid, then I have cells in my brain that are going to fire, right? And so I have that for each of these different hexagonal grids. And so when I stack those on top of one another, then I'm basically going to know my location based on where all of those different grids that fired all overlay the most. So that's kind of cool. So they're found in these modules, which are collections of cells that have similar spacing and orientation, but different offsets, right? So you could picture like some sort of grid paper, you shift it a little bit, but it's going to maintain the same spacing. So that gives you one module of grid cells. And these come in a variety of different scales that range from 20 to 30 centimeters worth of spacing up to meters worth of spacing, right? So that's kind of how we're getting at this local indoor kind of navigation versus like being able to navigate outside where things are a lot farther away. And so just to reiterate one final time, the overlaid firing patterns are what allow the brain to determine your location, right? Because if you had to program a single place cell for every single place in your environment, that would be very computationally expensive. Whereas if we have just these nice grids that overlay and stack nicely, then we're able to determine location. So does anyone have any questions before we get to the TDA portion?
Brittany, I have a question.
Yes, Alex, what's up?
Has this concept of grid cells been used in, for example, machine learning applications, or especially computer vision, and I guess, for example, like robot navigation, or in particular self-driving cars?
So that is an excellent question. And that question is something that the Army is actually very interested in. So I'll touch on that a little bit later. But just to give you kind of a preview, right? The Army is very interested in the ability of robots to navigate in space. As you might be able to imagine why. I am a public employee, so I don't know what they do with this. I just give them the math and they do stuff. So, right, a lot of our current machine learning systems are built on our visual system. So like the convolutional neural nets that you see oftentimes in machine learning are based on your visual. So if instead, like Alex mentioned, if we could instead build it off of something like this underlying structure, right, that would maybe give us better navigation purposes. So I have no idea if it's been used in self-driving cars, but it's definitely currently being used in machine learning algorithms. And we'll talk a little bit about that with the project that I worked on with Eddie. Great question.
I have a quick question. When we're talking about firing patterns here, like on an individual cell level, are we treating firing as like a binary state?
So that's an excellent question. So in this case, yes. So we're saying, okay, we have some spiking data, and what we're going to do is we're going to take that data and we're going to binarize it based off of time bins. So I'll talk a little bit about that pre-processing in just a second, but the way we can kind of initially think about it is that if I have a neuron that fires a whole bunch within this particular time bin, then I'm going to label that with a 1 and say that it's spiked.
All right, thank you.
You're welcome. Great question, Nate. Lucas, what's up?
Can you describe a little bit on the the picture you show here?
Yeah.
So yeah, basically what does the black points and then the red area means?
Awesome. Yes. So if we were, let's see if I can zoom with PowerPoint or if it's going to be stuck. Aha. Does that zoom on your side, Lucas?
Yep.
Okay.
It did.
So you can see this very light gray line that kind of traces under everything. So what that is that is the path of a mouse that it took through its environment over some recorded time session, right? So the sad part of this is that I love animals, but what they do basically is they go in and they attach all of these little probes into the mouse's brain that capture firing activity. And so they track the mouse's actual location. So that's that underlying gray path. And then they track when a neuron fires with a dark gray dot, right? So basically, as this mouse traversed its environment, anytime it hit one of these like kind of grid cell plots as we go up, right, that cell fired. And so they went ahead and put a black or like dark gray dot on it, right? So based on which neuron you're recording, right, this represents the neural data from one neuron, this represents the neural data from another neuron, et cetera, et cetera. And then they basically took that and then they did some sort of mapping in order to get a very pretty grid picture in like a sort of like data smoothing way to get these pictures below. So it's basically trying to capture this underlying structure that you see in the grids above. Does that answer your question?
Yes, thank you.
Awesome. You're very welcome. Y'all are asking great questions. I love it. Do we have any more before we continue on our journey? In that case, we'll continue on. Okay, so there is one last cell that we'll briefly talk about, and that is a conjunction cell. So these are found in the MEC as well. And each conjunction cell is just a combination, basically, of a grid cell and a head direction cell. So these conjunction cells fire when the animal is on a certain grid location that has also a certain head direction, right? So basically, it's just your brain kind of said, okay, well, this is kind of a nice way to take a few shortcuts. We'll combine it into one, call it a day. So why do we care about these cells? And Alex already asked a great question about why we might care. And our first level is kind of that we want to be able to decode neural data to reconstruct an animal's path, right? So if we're able to take just the neural spiking data and then say, okay, all of these neurons fired, the animal is right here, right? That's pretty cool. We also want to understand how the brain's navigation works, and we want to be able to recreate that into artificial systems. It also gives us some insight as to how the brain evolved, right? Because they've found these cells, certain kinds of these cells in things like mice, but they've also found them in primates and cats. And there's some evidence for human, but as you can imagine, human testing is a little more controversial than testing on animals. And so if we can uncover this underlying structure of the systems, if it exists, which hint it does, then we want to be able to utilize it. Okay, so the goal of the current work that I've been working on with my collaborators is to create a machine learning algorithm that takes in this underlying connectivity of the neural data and then is able to predict a head direction or a location. So we'll kind of break this down into a few steps. We'll identify the neural spiking data that we want to decode, and we'll talk a little bit about the topology of that data. We want to generate a simplicial complex from the neural spiking data. We want to feed that simplicial complex into the simplicial convolutional recurrent neural network structure. So for short, we'll call that the SCRNN. And then we want to output a head direction or location. So this is just the overarching view. We're going to go through each of these steps individually. So if you have questions, let's see if my slides answer them, and we'll go from there. Okay, so the first thing we're going to do, right, is if we're going to try to decode data, we want to actually have some data. So we're going to look at two different kinds of data. We'll do the head direction cells as well as the grid cells. So for the head direction cells, we have cell firing activity recorded for mice via probes in the brain for about 22 head direction cells. So the animals that forage in an open environment, and then the spiking data is recorded from all 22 cells. So you have the brain data recorded, and then the scientists also simultaneously externally record which way the animal is facing based on lights that are mounted to kind of its like headpiece as the mouse looks around. Also, I did not do any of this like animal processing information. These are all from papers. So the head direction data is from Peyrache et al. And then the grid data is going to be from Stensola and Moser, which I'm pretty sure this is the paper that is the big deal Nobel Prize paper. So the grid cell firing activity is recorded from about 200 cells, and these animals forage also in an open environment, and they record the firing patterns of cells for different levels of the MEC to get the different modules. So remember, the different modules are just those different grid spacings. Okay, so first we'll start with the structure of the head direction cells. And what's interesting about them, and what might not surprise you if you think about it, is that the activity of these head direction cells lives on a circle. So basically, right, we talked earlier about how clusters of cells fired for their preferred head direction. And so if we look over here, we have some sort of data that we can look at. So we have our actual head direction, which is this line in black. And then we have two predicted head directions, one from the cells that come from the POS system, and one from the cells that come from the ADN system. And so you can see, if we look closely at this red line, it pretty darn closely follows the actual head direction that was recorded of the mouse. So this specific time was for wake. These specific plots were for sleep. And also what's interesting is these head direction cells fire both when you're awake and when you're sleeping. That's kind of fun. And so the way that Peyrache went ahead and grouped and organized these cells was to stack them so that you kind of got some sort of artifact of the curve. But in reality, those cells do not live next to each other in the brain, right? Because remember, we talked about they were scattered all over instead of just being consolidated into one part. So for our work, we looked at the spiking data of 22 neurons over 20 out of 38 minutes of the foraging session for one mouse. And what that gives us is a very high dimensional 22 by 200 matrix of spiking data. So what we want to do is we want to take that spiking data and we kind of want to project it down onto a lower dimensional manifold. And so if we do that, the manifold that works the best is the circle, which makes sense because your head direction kind of can map to a circle because you can be facing 0 to 360 degrees. So similarly, the structure of grid cells lives on a torus, which that's kind of interesting, right? Because we saw that we had these like grid patterns. But the way I like to think about it is that it kind of makes sense because we could shift up our grid, or we could also shift to the right our grid, and we would end up getting kind of that same repeating pattern. So you get this kind of circular structure in two directions that allows you to capture that underlying connectivity. So this is also kind of a big deal. So this was outlined in a paper by Gardner et al., which I've gone ahead and cited below. And these grid cells can be split up into their modules, and the activity of each module can be mapped to its own torus. So what that means is that if I take my higher dimensional data, I map it down to a torus using some sort of mapping structure. So in this case, they did PCA to get down to 6 dimensions, and then UMAP to get down to 3 dimensions. So this beautiful torus that you can see with this line that traces all of these things, where trajectory is kind of what your color scale is based on. As your mouse traverses its environment, we can also trace out a continuous path on the torus. So that's how we know that this underlying structure is captured on this manifold. So that's pretty fun because it enables us to say, look at 482 cells, project them down to something we can actually visualize, and then see how those cells are related to one another. Any questions? Okay, we'll keep cruising. So our next step, and hopefully this will help a little bit, Nate, when we're talking about how we're processing the spiking data, is to generate our simplicial complex. So the first thing we're going to do is we're going to bin the spiking data into non-overlapping time bins. So right, we take all of our spiking data, we partition it up into even-sized chunks, And then we're going to generate the spike count matrix from that. So basically, if I have my neuron fire 3 times, I'm going to put a 3. And I'm going to do that for every single time bin for every single neuron. Then I'm going to do some row-wise thresholding in order to do my binarized matrix. So what that says is that certain neurons are going to fire more often than other neurons, just based on their normal activity. So we want to reference the neuron itself to see if it's firing a lot, instead of just referencing the entire firing structure of your brain. So we do this row-wise by just looking how much is this neuron firing compared to how it normally fires. And from there, we can come up with this binarized matrix, which will allow us to put a 1 anytime we have sufficient thresholding and a 0 anytime we don't. And then second to last but not least, then we can construct our simplicial complex. So the way we do this is we say, okay, I'm going to take, in this case, 6 time bins, and I'm going to draw a connection anytime 2 neurons happen to fire in the same time bin. So for example, here I draw an edge between my red neuron and my orange neuron because they fired here simultaneously, right? And then if I have 3 neurons fire together at the same time, then I'm going to insert a triangle into my simplicial complex, right? So that a lot of different neuron processing data things have used graphs in the past, right? Because you get that pairwise connectivity, but as you all know from doing topology, with simplicial complexes, you're able to capture that higher order connectivity. So last but not least, if we're going to do some topology, we probably need to assign some cochains. So for the first dimension, we're just going to go ahead and assign our spike count matrix number to our 0-dimensional elements. For the 1-dimensional elements, we're going to attach the Pearson product moment, the correlation coefficient of the appropriate rows of A. And then for 2-dimensional, we'll attach the multiple correlation coefficient of the appropriate row vectors of the spiking matrix A. So basically, right, we're leveraging this underlying structure, and this structure is what we're going to feed into our machine learning algorithm. Okay. And so, like I said, this is what we're going to feed into our simplicial convolutional recurrent neural network. So here is our overlying kind of framework, and we'll talk a little bit more about this upper right-hand portion, but I think it's nice to see it kind of all in one fell swoop. So we take in our neural activity, we pre-process it into a simplicial complex like we did in the last slide. Then we'll take that simplicial complex and we'll feed it into our simplicial convolutional neural network that you can see kind of the structure up here. And we'll talk about this on the next slide. Then after we feed it into our SCRNN, that's going to spit out in either head direction or location based on which data set we're looking at. And then we'll be able to compare that against our actual true state that we have recorded from the mouse itself. Okay, so I told you we were going to talk about this, so hopefully I'm going to hold myself to that. And so let's start with looking at the SCRNN. Okay, so we have our simplicial complexes input, and the way we're going to kind of input this into the network is as follows. So first we'll start by making the k-dimensional incidence matrix, which I'll say B sub k. So that just means if I have, 2 nodes that are connected by an edge, I'll insert a 1 or a -1 depending on your orientation. Okay, from this, right, that gives me some idea of the connectivity of my vertices, or my neurons is the way you can think about it. So with this connectivity, I'm going to then look at my Hodge Laplacian, which says, okay, I'm going to capture my lower order connectivity with B sub k, and I'm going to capture my higher order connectivity with B sub k plus 1, right? So if I'm looking at edges, for example, I want to know when nodes, I want to know when nodes are connected via an edge, but I also want to know when nodes are connected via a triangle, right? So you can go up at however many dimensions you need to. In this case, we're going to stop at 2 just for computational purposes. So we have our k-dimensional incidence matrix, which we use to generate our Hodge Laplacian. And then this lower line, this degree D simplicial filter, is what's actually giving us this machine learning framework. So we have W0 and WIs. Those are just going to be our weight matrices. So for any machine learning algorithm, right, what you're training is you're training the weights to kind of fire nicely. We have our k-dimensional incidence matrices. So remember, that's those B sub k's and those B sub k plus 1s, depending on what dimension we're looking at. In this case, D is just the degree we're choosing for our simplicial filter. And so basically, if I was to go through and actually calculate all of this together, this is going to give me my H sub k, which is my filter that I'm using for my convolutional layer. So I'm taking in my simplicial complex, feeding all of that into these simplicial filters, which I can do in multiple decked layers, right? Just like any machine learning algorithm, I can attach a bunch to each other. And then eventually that will give me out a flattened feature vector, which then I can feed into a recurrent neural network. So this is kind of the meat of the whole project. So if you have any questions, I'd be more than happy to answer them.
So, Brittany,
Yes.
so is it the same neural network that predicts both head location and or head direction and location? Or is it two different?
So that's a great question. So right now, the way we're doing it is we just have two separate networks. So we have one that predicts head direction, one that predicts location, but they're still utilizing the same architecture, which is why I threw them in the same slide. But that's a great clarification question. And it's actually, you're too good, Alex. You're leading me into future work that we'll discuss in just a little while. Okay. Well, if there are,
I have another
Lucas.
question.
Yeah, what's up?
So I think a few slides ago, you mentioned that there's a function that can match to high dimension data to, I don't know, like a
these ones?
3. Like, can you give me like a short, like brief summary of what the function is doing?
So the function that takes this high dimensional data and maps it down into lower dimensional space. Is that what you're asking?
Yes.
Okay, awesome. So I'm kind of using two different approach. Well, Gardner et al. Used two different approaches, which is how I've been thinking about this. So the first one says, okay, if I have my matrix, which I think is best talked about for head direction, right? I have this 22 by 200 dimensional matrix. So what principal component analysis does is it says, okay, I'm looking at my data in space, right? If I have, for example, in 2 dimensions, right, I have kind of an oval of data, then it's going to capture the vectors that best describe that data. So if I have this long skinny oval, right, I'm going to get a vector here and then a short vector here, and that's going to kind of reduce that down dimensions, right? So if I have like this like conical oval thing up here, and I'm trying to reduce it down to 2 dimensions, well, it's going to capture maybe this vector because that's telling me most of my data is along this vector, right? So I'm taking my 3-dimensional data, mapping it down to a single vector, right? So it's kind of doing the same thing now, just with higher dimensions. It's saying, okay, I have this like 22 by 200 dimensional matrix. I'm going to go ahead and just reduce it down to maybe it reduces 22 rows, 200 columns. So I want this to be a 22 by 6 matrix instead. So, right, it's going to do kind of that same process using a lot of linear algebra. And these are packages that I used in Python. And UMAP is a manifold mapping algorithm, which says, okay, I'm going to kind of do something similar, except instead I'm going to look at trying to identify like underlying structure in my data that's not just given to me by vectors, but maybe it's given to me by some sort of manifold, like a circle or a torus. And so I can give you some links to those different packages if you're interested, because they're really fun to play around with.
So when you're working on this pipeline of generating from the data to the topological mapping, are you solely use Python, or do you have other, or do you need other programming languages to
Gotcha.
set up the system?
Yeah, so the SCRNN was done entirely in Python. And it utilizes, let's see, I think Eddie used TensorFlow as the like actual machine learning portion. And right, the PCA and UMAP are used for the actual like manifold mapping portion, but those are also implemented in Python. And I think I have a link to the GitHub at the end of this. And if not, you're welcome to email me and ask.
That answers all my questions. Thank you.
Awesome, you're very welcome. So we'll go back and we'll go one slide further. So as soon as we have, right on the last slide, we had our SC portion, which is doing the simplicial convolutional stuff. And then we have the RNN portion. So our recurrent neural network is just employing the Elman recurrent neural network architecture, which is probably the one, if you've heard of RNNs, you're the most familiar with, where it takes in the data, feeds it back to itself, and then keeps feeding it forward. And then at the end of the day, depending on the data you fed in, it's either going to spit out the appropriate head direction or the appropriate location. Okay, so let's look at some results, because that's always fun with actual applied math, is you can throw stuff into a computer and see what happens. So we compared the SCRNN against 3 other networks, and we used 3 different errors to judge these networks. So MAE, it stands for median absolute error, AAE stands for average absolute error, and CAT stands for catastrophic error. So catastrophic error just says, okay, how many times was my head direction estimation off by 90 or more? Right? Because if you're off by 90 or more, your head is definitely facing the wrong way, and you have no idea what you're doing. So the R network, which again is the SCRNN, did pretty darn good. It had the lowest of all 3 errors, and it only had 6 catastrophic errors, which if you're, again, trying to navigate a drone in some sort of new environment it hasn't seen before, that's not too shabby. And so you can see down here, we have 2 different plots. So the first plot is just a minute subset of the data, where the true head direction is this dashed blue line. And you can see that our decoded head direction, this red line, follows it pretty closely. We have the occasional bad spike, so like here or up here, and it doesn't quite go as low down here as it does for the true head direction, but it's still not too bad. And then for the location data, when we took the same architecture that we used for the head direction, but instead we fed in our grid cell data, then we were able to do the same kind of process where we have the true head direction in the blue dashed line and the decoded head direction in the solid red line. And you can see that it actually traces out this mouse's path pretty darn well. And in some points, it actually seems like it's trying to actually predict the next one instead of the time step that it's currently on. And we do all right with our AED, and that's so for our training data, we're only off by 0.02 meters, and for our testing data, we're off by 0.15. So there's definitely some room for improvement, right? Because that's not the world's greatest error, but as a starting point, we were pretty content. Okay, so future work, and just a few last things to wrap up before we can do discussion and more questions. So we want to look at more applications of the SCRNN, such as applying it to other types of brain data like EEG, right? Stuff that you can look at externally without having to put a bunch of probes into the brain. We want to look at embedding this architecture into location-focused artificial intelligence. So kind of what Alex was talking about, maybe using it in things like cars, or some sort of other unmanned vehicles, and explore the different underlying manifold structures and how they all interweave together. And so this last one is my current project. And so I wanted to give you just a quick little idea behind this, and that is, right, we have these conjunctive cells that predict both head direction and grid location. So if I know where I'm, if I know which grid is firing and I'm firing on that particular grid, then I could also have a cell that says, oh, I'm firing on that grid specifically when my head is facing straight forward, right? So that's a point in a 3-dimensional space across 3 circles. So I have the circle given to me by my head direction, the circle given to me by one direction of my grid cell, and then the other direction of my grid cell. And so what I'm working on is trying to show that these manifolds that have been shown as separate are actually together, right? So instead, I'm looking at maybe for a single module of grid cells, right? This would give me a 3 torus. And so if I have a point on the 3 torus, that's going to give me a corresponding head direction and grid location. And how all of those different modules are going to combine is an area that I'm currently investigating. So selected bibliography, here's some of the sources that I talked about in my talk today. And last but not least, I'd like to thank my collaborators. So Edward Mitchell, who's the lead author on this paper, David Booth, and Piotr Branazic, I think is how you say it. There are ARL collaborators, and then last but not least, my postdoc advisor, Vasilios Morales. And so this work was funded by a US Army Research Lab contract. As you can see, the contract number is given below. And if you enjoyed this talk and you're curious about chatting more, you can contact me at my UTK email, or I also have a website that you're welcome to go look at. So thank you all so much for your time, and I'm more than happy to take any questions that you have.
Yeah, thanks so much for the talk, Brittany. It was very, very nice. Are there any questions for Brittany?
So, yeah, I have one. Can you go back to, I guess, the slide on predicting trajectory? Yes, I believe this one. Yeah. So, yeah, we see that it does do a really good job, I would say, of tracing out the trajectory, but it sometimes like the decoded path takes these weird turns and briefly goes off in like the wrong direction. Yeah, like in there. So do you think that studying, these conjunction cells would be able to mitigate this effect, since it's taking head direction into consideration at the same time?
I think that is an excellent question, right? Because as an animal is walking, I would say, when you're walking around, most of the time your head is probably facing the direction you're walking. So I think having some additional information definitely seems like a good way to approach the problem. So I think, if I remember correctly, when Eddie worked through this data portion, he was looking specifically at the just pure grid cells. So I think that would actually be a really good approach, is to take the head direction into consideration as well. So my answer is we haven't done it yet, but it's a great idea.
Thank you.
Of course.
So yeah, I have a question, Brittany. So in like an animal's brain, like, could you, like, let's say you cut open an animal's brain, could you, like, I guess, how would you go about finding one of these grid cells or head direction cells or something like that? Like, is there, I know they're located in a specific area of the brain, but like, are all of the cells in like those areas of the brain grid cells, or right? Like, yeah, I guess, how do you go about finding these?
So I would like to preface this with, I am not a biologist, and I highly recommend you talk to someone who is a proper biologist and neuroscientist about this. So this is just my interpretation of what happens. So there have been a lot of studies over the years, especially with like probably lower taxonomic order animals, right, where you can stick a bunch in the brain and you can just, as they do things, you record the firing patterns and you can start to draw associations that way. And so there have been enough studies done over the years that they have a pretty darn good idea of where all of these things live, at least with the animals that they most commonly use for testing. And so they can insert the probes and like kind of get like a stack of neuron recordings as like it goes along that probe. And so you're able to capture that information. I would say that not every one they hit is probably the grid cell location, but they're dense enough in there that you can actually record them properly, is how I would phrase it. And again, this is just my understanding. For more information, all of the sources that I listed that are actually neuroscience sources are a great place to start. Especially, I feel like this one the Moser paper does a really good job of kind of explaining all of the underlying sampling that goes on, because the data came out of that lab.
Okay, great, thanks. So yeah, are there any other questions for Brittany?
Okay.
I have a